and her husband. I actually go for it. I told him. Yeah, but she had tickets and her husband works early the next morning, so he's like, I'm not going. So guess who's going? Hey, you get to see Posty. Hey, I get to see Jelly Roll.
Me too. Honestly, I can't do it in one sitting. I have to pick it up.
Yeah.
I just saw a bunch of TikToks about them this morning.
I can look Jelly Roll.
Alright, let's get started. Oh, it's still not working. They must have not come to fix it, or they're waiting on a part. Sorry about this. Alright, go ahead and adjust your seats if you need to. Looks like we're doing this again. Alrighty, let's discuss our last day of genetic talk, and then next class we're moving on to metabolism. It's a nice change of pace for a bit. All right. Okay. So we've talked about how we copy our DNA and how we convert the DNA code into proteins, how we modify those proteins and all of that. But the question then is, so is this just happening all the time to every single gene? Do we constantly express every single gene? What level do we express these genes at? So that's what we're hitting at today. But basically, we don't. That's the short and sweet of it. We want to have extreme control over what genes we express at what time and in what quantity. Because sometimes we don't want to express heat shock proteins because we're not experiencing heat shock. We're happy. So for a number of reasons, we don't express every genes, basically like physical space. We can't constantly have all those transcripts being made and all those proteins. It costs a lot of energy. So why am I going to make a protein if I don't need it? And we can have a protein that does one thing and another protein that stops that protein from working, and we don't want those contradictory functions. Sometimes we just want one protein to work and we will wait to make the contradictory protein until it's necessary. Alrighty, so we can alter our expression levels at many different points. We can alter the DNA sequence, like with mutations. This is the most permanent, it's the most extensive, it's the one with the biggest consequences. So we tend to not do that one too often. One of the main ones actually are these two right here, control of transcription and control of mRNA stability. If we want the mRNA to be copied more, we need to make it more stable so it has a longer half-life. If we don't want to make a protein, maybe we stop transcription and we block its ability to even turn into mRNA. And then we also have translational control, where we made the transcript and we're translating it, but maybe we don't want to translate, tons of copies, just a few. So we have lots of different ways, including also post-translational control, so we can actually then take the proteins afterward and modify them to either block them or do something else with them. So this is what we're going to be discussing, and we're mainly going to focus here in this transcription and RNA stability discussion. We'll get a little bit into translational control, but for the most part, we're staying at the mRNA level. Any questions? All right. So the first thing we have to understand, and I realize now that I've been throwing this out there a lot, and hopefully you guys have had some type of genetics or something so you understand this a bit, but I've been saying, oh, it's a repressor a lot, and we haven't really gone over what that means yet. So when we are initiating transcription, we need to attach our RNA polymerase and our sigma factors, and we want to have a high control over that. So there are other proteins that can bind to the DNA that affect that whole process. Either they make the RNA polymerase and the sigma factor bind better, or they prevent them from binding at all. And we call those repressors and activators. And changes in the environment within the cell or outside can also affect the function of these proteins. We can have like an antibiotic that can bind to one of these proteins and prevent it from binding DNA at all, or cause it to bind the DNA. So there's lots of different environmental factors and cues that will affect these two types of proteins. Have you guys heard of these before, repressors and activators? Most of you? Okay, cool. We're still gonna hit them, but it's nice to know that at least we've heard, this term has been out there before in our brains. All right, so these are called regulatory proteins because they're regulating when we make our mRNA and when we don't. So repressors are going to bind the DNA at the promoter. Activators are going to bind the DNA near the promoter. Because the idea is a repressor wants to stop us. It wants to stop RNA polymerase from attaching and making that RNA. So what better way to do it than to actually physically block the space where RNA polymerase would bind? An activator wants to make us make this RNA a lot more. So we don't want to block where the RNA polymerase will attach. We want to bind near it to encourage the polymerase to come and attach to the right place. So that's why they tend to have slightly different binding areas. Now, they have an interesting structure, and they're often, we mentioned this before, I believe, they're often in the form of dimers. So they will bind the DNA at two very specific sites. Usually they'll look for a specific sequence in that major groove of the DNA, and they'll bind there. And often, we have, the dimer is, here's one part of the protein, here's the other, and they're basically like mirror images of each other. And often then we will have a second one of these on the other side of the DNA as well. And so we call that a tetramer or a dimer of dimers. And that's very common in bacteria for us to see these types of repressors. So any questions about that? Right. Now, what is the DNA that they recognize and bind to? Well, we call these sequences inverted repeats, meaning they're going to have one code that is read in one way, the same as it's read in the other way. That way, wherever this protein is mirrored, it can bind in the opposite direction. Because since we have the dimer, in one way that it binds, it's going to be able to read, let's say, left to right. But then we turned it in the mirror image on the other side, and so I can't read left to right anymore. I have to read right to left. So my repeat needs to be flipped as well because of that. So the sequence is just a repeated sequence that's flipped to mirror itself. That's the idea here. And the sequence is going to affect the affinity of binding. So affinity just means how likely and how tight it is to bind that sequence. If you want this regulator to bind very, very tightly, you want a very high affinity. You want the structure of the protein and the sequence of DNA to make a super tight fit with lots of connection points, because we don't want that to fall apart and we want to encourage that binding. But if this is a gene that we necessarily don't want blocked all that often, or we want to be able to turn it on more frequently, we might have a lower binding affinity where it'll bind, but it'll break off pretty easily too. And the more similarities, the tighter the binding. Any questions about that? Sorry, I have to get back into the teaching today. It was a very, very, very long weekend. All right. So how do cells know how to regulate their DNA? Remember, we are kind of personifying these processes. They don't know anything. They don't want anything. They don't plan anything long term. They're responding to chemical signals in the environment. And that's hard for us to understand, so we tend to personify it to make it make a little bit more sense to us. But there's intracellular and extracellular cues that they can respond to. So intracellular cues are often metabolites. So we're doing metabolism and we make a bunch of intermediary products or waste products from it, and those will often bind to our repressors and our activators because most often the cell wants to change its expression regarding to how it can eat and how it gets energy. And we call these, if they're binding to our regulatory proteins, we call them ligands. That's a term we've heard before, right? If you remember from back in the day when you were first learning about enzymes, enzymes have an active site, and that's where they bind the substrate. Basically, a ligand is a substrate. It's just the thing that binds to the protein to the enzyme. Any questions about that? The key here is whatever the cell is sensing, it's sensing it from inside the cytoplasm. These are chemicals that are dissolved in the cytoplasm, they're floating around, and they're binding to these repressors or these activators. Now, let's go over how this works. So how do I want to go about this? Sorry, this doesn't have a good, I thought this had a better transition than it did. All right, so basically these proteins are going to bind to the DNA and they have their affinities for it. The ligand is going to affect the affinity of binding. Either it's going to bind to this protein and make the protein shape alter just a little bit so that it binds the DNA more tightly, or when it binds, it's going to alter the shape of the protein so that it falls off the DNA more readily. So that's the main idea behind these. And whether it causes it to bind more tightly or loosely is highly dependent on the protein and the metabolite and all that good stuff. It's case-by-case scenario. Sometimes we even want a repressor to bind more tightly, or we want it to not bind tightly, so it can have different metabolites that cause different responses on the same repressor. So let's dive into that a little bit more. We're going to start with repressors because I feel like they're just a bit easier to understand. They are going to prevent gene expression or repress it. That's what we mean by that. They're going to come in, they're going to bind the promoter, and they're going to say, RNA polymerase, halt. You cannot bind here. We don't want this mRNA to be made today. Now, some of them will bind whether or not there's ligands around. So they just find these sequences and they bind them no matter what else is going on in the cell. Others will bind only after they're bound to a ligand first. So they're floating around and they can't bind to the DNA until one of these ligands come, they connect, and now it can bind to the DNA. So we call that ligand an inducer. It's something that induces the repressor to bind. Inducing just means encourages or turns on. We kind of use it in both ways here. A co-repressor is a ligand that will bind to the repressor and enables it to bind to the DNA. So the co-repressor is the one that comes in, binds to the protein can then bind to the DNA better. It wasn't able to bind before. The inducer comes in and says, oh, you're already bound to the DNA, but we want to induce this gene. We want to turn it on, so I'm going to bind to you and remove you from the DNA. And now we've induced or turned on gene expression, encouraged gene expression. So I'm sorry if I said that backwards. I think I might have. So when we induce, we want to induce the gene expression. We want to turn it on. So the ligand comes and binds to an already bound repressor and removes it. A co-repressor is like a coworker. They're going to work together to bind the DNA and stop expression. They're repressing together, basically. Have you guys heard this before? I'm glad that we have a lot of repeated knowledge for some of you, because that makes this exam a little bit easier, right? All right. Yeah, fingers crossed. Okay, activators, on the other hand, want to stimulate gene expression. We are saying, let's turn this on and let's make mRNA. We like this gene, we need the protein from it. And so instead of binding to the promoter, they bind near the promoter, and they're going to encourage the polymerase and the sigma factors to come in and bind, and we'll talk about how they do that. Now, usually when they're bound to this sequence, they're very loosely bound. They don't bind very tightly on their own. They need a ligand inducer to help them bind more tightly. So they'll bind and they'll encourage expression unless that inducer is around, and now they're like, okay, foot on the gas, let's go, we're bound tightly, we're gonna get to make this a lot now. Because if it's not bound very tightly, it's more likely to fall off. And so it won't be able to encourage that expression as frequently because it's constantly falling off. The inducer keeps it attached. Make sense? You guys are like looking back at me the way I feel right now. All right, so let's try to make it cool and I'll try to be as upbeat as I can. All right, now, when we want to respond to an external threat or a food source, we can't just rely on things inside the cytoplasm to bind to these proteins. We have to somehow overcome the barrier of the cell wall. So if there's a food particle like lactose that's outside of the cell, we need to be able to know it's there to turn on the genes to bring in the lactose. Or if there's a threat like an antibiotic, we need to make more pumps to pump out antibiotic, or we need to turn on our flagella to swim away. So how do we sense those signals outside of the cell? Well, you guys have heard of signal transduction, right? The secondary messengers like CAMP and the kinases, basically the same idea. We have a two-component signal transduction system in bacteria. There's many of these, not just one. Just like it with eukaryotes, there's many signal transduction cascades that can be triggered. The way these work, there's a kinase in the membrane that can recognize the signal, and kinases, all they do is they just phosphorylate things. They add phosphate groups to things. So when they bind the signal, they phosphorylate a secondary messenger, which then binds to the regulator, the repressor or the inducer, or they cause a signal transduction cascade, and then whatever the end product is binds to the repressor or the inducer. We're not gonna need to know that whole deal. You learn that in cell bio. You just need to know that they have a similar mechanism, and it's called a two-component signal transduction system, and this helps the regulators respond to external signals. That's the main takeaway I want you to get from that one. Cool. Alrighty then. Let's keep going. So this is just kind of saying what I just said. There's the kinase, it responds, it phosphorylates, and we call it the response regulator. That's the cytosolic protein that's stimulated. That's just a little bit more to that. But you don't need to know more than that. This slide is just a way of saying, okay, that was a lot of information. Let me put it all in one slide to summarize it for you. So that's there to help you when you're studying. Trying to do that for these difficult concepts a little bit more now. All right. The lac operon. This is going to be the big guy today. This will be on the exam. This and the trip operon. If you plan to take bacterial genetics, you got to know these guys. It seems difficult on the surface, and you want to be overwhelmed by it, but I promise you, when you pause for a moment and breathe, it's very direct, and we'll make it as simple as possible today. So this is one of the things that I was told, make sure they know it. So it will be on the exam. So let's go slowly and take our time with it, because other things I can cut out, this you have to know. So any questions, please stop me at any time. So this was first found by a couple French scientists in E. coli. Now, we found similar things in other bacteria, but we study E. coli more. And so this is the model organism, and it helped us figure out these regulation patterns in other cells, including our own. So it was kind of the template that we learned to then explore other places. Now, they found that the enzymes that would eat up lactose or metabolize lactose, those enzymes were inducible. The cell could turn them on at will, depending on whether there was lactose present or not. And then the enzymes that would metabolize glucose were what we call constitutive. They are always turned on, they are never turned off. So what that tells us, just to get in the right frame of mind for the lac operon, glucose is the primary food, whether you're a bacterial cell or our cells. We prefer glucose. That's why we always want sugar, especially late at night. We want that sugar to help our brain work better. So everyone's preferred form of food is glucose. But what if there's no glucose around? What if we ate it all? What if we're in an area that we don't have any glucose? Well, then I want to be able to turn on genes that let me eat other things. That's the idea behind this. We're going to always eat glucose, but what if there's low glucose, or what if we need more energy? Well, we need to turn something on to eat this other sugar. One thing I do want to say in advance, because it's very easy to forget, we are multicellular organisms with digestive systems and body systems. They're just cells. So we're talking about a cell response, not lactose intolerance. That's a disorder where we don't produce a proper enzyme to break down lactose. This is different. I know we want to think, oh, the cells are lactose intolerant. No, that's just something that we do to try to make it familiar to us. All right, so we're going to look at how do we turn on this gene, how do we regulate it, and what happens if there's high glucose, high lactose, high glucose, low lactose, low glucose. We're going to look at all these conditions here. And I want you to not so much memorize this, but try to understand it and ask yourself, okay, if there was a lot of glucose and only a little bit of lactose, what would happen? If there was low of both, what would happen? If there was high of both, what would happen? So don't just list off things that you know. Try to explain what's happening for the exam. That's my tip for that one. So let's dive into it. There is a repressor called LAC-I, and that one is going to repress the whole lac operon. Because I feel like the book never really directly lets us talk about this, it doesn't sit down and says, let's talk about an operon. Just to be sure we're all on the same page, we talked about how our mRNA could be polycistronic, meaning many genes in one mRNA transcript. So we'll have the promoter, we'll have the operator, which we'll talk about today, we'll have gene 1, gene 2, maybe even gene 3, 4, 5, and then we'll have our terminator sequences that kind of end it all. This is what we're talking about when we say an operon. It's the regulatory aspects and all the genes associated with it. So we are talking about a repressor that is repressing a whole set of DNA and genes that's involved in eating lactose, not glucose. So this is what we're working with today. They don't have introns and exons, they only have operons. So it's a slight difference between our DNA and their DNA. All right, so the repressor is LAC-I, and we have an activator of cyclic AMP, CAMP. You've probably heard of that one before. That was the secondary messenger for signal transduction in eukaryotes. So we're making connections here. It's also a secondary messenger in all cells. Now, we're going to be talking about this in low transcription versus high transcription conversations. In the end, no gene is ever 100% completely turned off all the time, because that's just not physics possible. Because all this is, if we're turning off a gene with a repressor, all it is two molecules bound to each other, preventing that from being copied. But in reality, those won't stay bound together forever. Just the natural movement of molecules that will eventually fall off and will sneak in a couple transcripts, and then it'll bind again. So we're never talking about fully off and 100% on. We're talking about, it's kind of squeaking by a little bit, to we're gonna ramp it up to whatever we need it to be. So this is a toggle switch, or a rheostat, and not an on-off switch. we ready to go? This stuff's fun, actually. It seems stressful at first, but I promise if you try to look at it from like, this is really cool that cells know how to do this, it makes it a lot more interesting. Let's talk about lactose first. What is it? What structure is it? It's a disaccharide, di meaning two, saccharide meaning sugar. So we have two sugars, two simple sugars pulled together and connected by a covalent bond. And those two sugars that are connected are glucose and galactose. So glucose and galactose come together and make lactose. Now it can be used as a carbon and energy source. So we can break it down, we can get food from it, we can get our carbon from it to make other molecules, we can do a lot of things with it. But it requires two things in order for us to be able to use it. A protein called lactose permease, the gene for it is LAC-Y, the protein is called LAC-Y with a capital L, and beta galactosidase or LAC-Z. So let's rename these genes here. We've got LAC-Y now, and we have, I believe it's over here, LAC-Z, and there's another one in the middle that we're gonna talk about, but we don't really know what it does. So we don't really talk too much about it, we just say it exists. So these two proteins are gonna be the main part of how we take in lactose and eat it. Now LAC permease is a membrane protein, and it controls whether lactose is allowed into the cell or not. So it's just a little transport protein that's gonna capture the lactose and bring it in if it's available. Beta galactosidase has actually a couple different functions. It breaks apart the lactose into glucose and galactose. It'll break that bond between the two sugars. It can also take the lactose and modify its structure to turn it into something we call allolactose. So it has a lot of different enzymatic activities, but the main ones turn it into allolactose or break it apart. Good? Have any of you heard of that background before? Oh, yay. Okay, good. Whew, I was like worried. I'm like, oh no, they're never going to have heard this before. We're learning it all from the beginning. That's okay if you are. That's 100% okay if you are. But it just helps me breathe a little easier today. All right, so when we want to bring lactose into the cell, we use that LAC-Y, the lactose permease, and we bring lactose in while also symporting a proton. So it's powered by proton motive force, which we're gonna hit pretty hard in the next couple days with metabolism. But basically we use that movement, that flow of the protons, the hydrogen ions, to power the movement of lactose into the cell. Then beta galactosidase takes that lactose right here and it converts it into allolactose. And allolactose is really the signal that helps turn on this whole operon and make it work better. There's always going to be a tiny amount of beta galactosidase inside the cell, and that's why if any lactose comes in, we can convert it into allolactose and turn on the whole operon, because there's always a little bit around just in case. It's our way of sensing, is there any lactose? Otherwise, how would the cell know if there's any lactose to turn on this operon? So it always makes a little bit to know, okay, we got to turn this on a little bit more because now we need to eat lactose. Any questions about that? All right. Now, oh, I totally messed this up. Here I was thinking, oh yeah, I remember the order of these. Sorry about that, guys. So we have LAC-Z at the beginning, then we have LAC-Y, and then LAC-A. Yeah, the LAC-A, we know what it is, but we don't really know what it does. Like, why does it have to be turned on with the rest of these? What role does it play in this whole process? We don't really know for sure. So we have a name for it. It's thiogalactosidase transacetylase. That's it.
That whole name.
No, I won't make you know that name. Don't worry. Yeah, a lot of sciencey words there, you don't have to know. All right, so the idea is all of these genes are controlled by one promoter and one operator sequence. So once the promoter gets turned on, we make all of these transcripts, and then therefore we make all these proteins as well. That's the idea here. The operator sequence we call LAC-O. The promoter sequence, we label it P-LAC-ZYA because it's the promoter for genes LAC-ZYA. It's nice when they all have the same base name and it's not like LAC-Z, BIC-Z, and you know, all those funny things. Alrighty, let's dive into this then. So LAC-I, our repressor, is encoded by a gene that's elsewhere. So it's not, we don't see LAC-I here, which is kind of important. I want to be able to control the repressor separate from my gene because I don't want to have to turn on my gene to repress my gene. That creates problems. So if we continue our DNA, what we end up seeing is there's a promoter here and there is LAC-I over here, and it's going to be transcribed in that direction. This is going to get transcribed in this direction. So we say it's upstream because it's going to the left of our gene, and divergent, or going in the opposite direction for transcription. If you hear those terms, that's just what they mean. Now, this LAC-I is constitutively expressed, just like our glucose metabolism, meaning we're always making a little bit of it in the cell. It's always turned on. So that means we're always going to have it binding to the promoter and operator and blocking transcription of this unless we forcibly turn it off, unless we make it disassociate. So you see here, this is the code. We're going to talk about our activator later, but this is where the activator would bind. This is our promoter with the -10 and -35 consensus sequences for the whole operon. And then overlapping our promoter is where the LAC-I repressor binds. So if it's first overlapping with our promoter and it's also ahead of it, even if the RNA polymerase was trying to bind, it can't because it's being blocked. But let's say it could bind just a little bit. It tries to move and it's blocked by a physical protein sitting here. So we can't get to the start to start making our gene. Does that make sense? Okay. So let's keep going. Just another way to see that. I wanted to show you the overlap with their binding sites, but also if you need to see it on the gene itself, there you go. The repressor is made here, and it's binding to the rest of it over there. All right. So in the absence of lactose, so there's no lactose around, LAC-I is going to bind as one of those dimer of dimers. It's going to have two proteins come together to form the dimer, and they both clamp down on the DNA. When they do this, they form a loop structure in the DNA itself. So now, how the heck can RNA polymerase bind there? No way at all. Not only does it have a physical thing blocking it, but it has this weird structure in the DNA that it doesn't know how to bind to. So we have fully blocked our sigma factor and RNA polymerase from binding. And it prevents that open complex. So I'm going to start trying to make a table here for us to think about. So low lactose, we have LAC-I bound to promoter. No transcription. That way we can kind of keep a tally of where we're at. Now, in the presence of lactose, we sense that lactose is coming in and that beta-galactosidase is doing something to it. We're going to have a de-repression event. We're going to remove the repressor and induce this whole operon. Induce means turn on. So when we have lactose come in, beta-galactosidase will turn it into allolactose. Some it cuts, some it turns into allolactose. Like we said, it has those two jobs. Allolactose binds to our repressor. It acts as a ligand that binds to the repressor and pulls it off. We call it an inducer. So, when lactose is present, we're turning on the gene, which makes sense. It's like, hey, there's a food here. There's ice cream now. Let's make spoons so we can eat the ice cream. Before we were just making forks and knives. Now we need the other things to eat all the other food. So when lactose is present, the beta-galactosidase, I'm just going to call it beta-gal, is going to convert the lactose into allolactose. That allolactose will induce the operon by binding and removing lac-I. Sorry, that is super messy writing. I'm just trying to get it done so we can move on. I'll try to write a little neater next time. So, no lactose, we want the repressor to be bound. But now lactose is around, we want the lactose basically to bind to our repressor and pull it off so we can start making more of what we need to eat the lactose. That's the idea behind that. So this is just a review of what I just said here. Now, let's talk about how we activate this whole thing. So we don't necessarily, we want to be able to say, okay, lactose is present, remove the repressor, let's make this. But what if we wanted to really, really make this? What if we're saying we have no glucose and we're starving and there's lactose around, eat the lactose as much as possible. We want to be able to turn up that rheostat. We want to make it louder and brighter in here, make more enzymes so we can eat the lactose more and more and more. So we don't just want to be content with, well, it's on, shouldn't we just be fine with that? No, we want as many spoons as possible so we can eat the ice cream as fast as possible, basically. And here's where CAMP comes in, cyclic AMP. So this will accumulate when a cell is starved for carbon. Basically, when we eat up all the glucose, this CAMP will start to form to tell the cell for its starvation mode. Just like we learned with that two-component toxin system, the toxin-antitoxin, where we have PP, GPP, and CAMP start to accumulate inside the cell, it's a signal that we are starving here. So the CAMP accumulates, and it's going to start interfering with how we look at our operon. There's a caveat I'm going to say here. Textbooks haven't, for some reason, quite caught up to this yet, and I think it's because we don't want to confuse students. We want you to learn the idea behind this here. But in the 90s, they found out that CAMP does not increase when glucose is gone, if lactose is present. Because the cell says, well, glucose is my favorite food, but lactose is my second favorite food. So if there's still lactose around, I'm not starving, so I'm not going to make CAMP. So it was something where we thought we understood this at first, but now there's more information. CAMP still does do everything we're going to say, but it doesn't necessarily turn on its highest just because there's no glucose around. So keep that in the back of your mind. We're learning the perfect version of the model that helps us understand the idea of repression and induction and activation. We don't care necessarily about the actual details of this. We just want to understand it enough to know, how does repression work? How does inducing and removing the repressor work? How does activating work? That's all I want you to get out of this, okay? But just keep that in your mind, that there's a little asterisk to this whole discussion that we're having. Okay, so let's keep going with this. So CAMP doesn't just control the lac operon, it controls the expression of many, many genes, which makes sense. If it's an alarm molecule that gets introduced into the cytoplasm, we want it to be able to affect any gene that it needs to respond to that alarm. So in this case, there's going to be a binding event. CAMP is going to bind to the CAMP receptor protein, CRP. Some textbooks also call it CAP, CAMP acceptor protein. You can use whichever term makes sense for you. I'm pretty sure the book uses CRP. So that's what I'm going to say. If we want to turn on the lac operon as high as it will possibly go, make as many of these genes to eat lactose as possible, we need CRP. We need CAMP to bind to CRP, and we need it to bind to the activator site, which is going to encourage the polymerase to bind and make as many copies as it can. So basically what this is saying is we are starving. We form this CRP because there's more CAMP because we're starving. So let's turn this on as high as we can go so we can get as much energy from whatever is available out there. But this is predicated on the fact that lactose is also present. This necessarily won't do much if the repressor is still bound. So we have to have lactose in order to turn this on to its highest volume, because otherwise why would we do that? We'd be wasting the energy when we're already starving. Okay. Any questions about that? Cool. So it does this cool thing. When CRP, the CAMP CRP complex binds, it causes the DNA to kind of bend and make this interesting shape. And it allows the CAMP CRP complex to associate with RNA polymerase and make it bind more tightly. Because in the end, if we want something to bind more tightly or loosely, it's all about changing its structure just a little bit. And we do that by binding or removing things from it. So if CAMP CRP can touch and associate with our RNA polymerase, it'll change its structure just enough that it'll attach to our DNA tighter, and it won't fall off, and we'll start making more and more of those proteins. So that's the idea there. It's going to bend it so it can associate and touch the polymerase and get it to attach better. And that activates the gene transcription. So the idea here is, I'm not even going to say low lactose or low glucose. I'm going to say starvation, so when we're starving or there's high CAMP, the CAMP CRP complex forms, binds to the activator sequence. I'm going to abbreviate some words here, sorry. Causes RNA pol to bind tightly. And this is to maximize expression. So this is low or no expression. This is medium levels of expression. And that is maximum levels of expression. So that's how we can kind of modulate it. Because in the end, if there's just a little bit of CAMP, it's gonna a little bit do this. If there's a lot of CAMP, it's gonna do this more and more and more and more. So as the CAMP levels increase, we're gonna express this more and more and more and more. Does that make sense? So in normal conditions, we don't want to use lactose. We'd rather use glucose first, so we turn it off. But if we want to use lactose because it's around, we want to be able to remove the repressor. If we're starving now and lactose is around, we want to make as much of this as possible to eat as much lactose as possible because we just need to eat. It's the idea of if you can't find a place that has salad or, chicken and steak and all the good things for you, a vending machine will work fine in a pinch. Any questions about this so far? All right. So if glucose and lactose are present, we get this really interesting scenario. And it's called metabolite repression. And it's the idea of, well, if you have the healthy food and the junk food, we should eat the healthy food first. And the cell is good. It will eat the healthy food first. And it'll save the junk food for later. So in this case, when we have glucose present, it's going to function to kind of turn this whole thing off and say, no, 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 you can rest for a while. We don't need you right now. So in this case, glucose is going to basically turn off this right here. So we bring it back down to low or, middling levels of expression for the lac operon. Now in the cells, this causes something really cool to happen, and you can see this very clearly on some growth curves. It's called dioxic growth. The cell will preferentially eat all the glucose first, and then it'll be like, okay, now I ate all this stuff, I gotta start eating this other stuff now. And so we'll see an exponential growth phase followed by a lag phase in the middle of this. Because now we're changing our expression. We were expressing everything we needed to eat glucose, but now there's no more glucose, so I need to alter and convert to these two forms and turn on the lac operon, and that takes a little bit of time. So there's a lag period due to the change in expression levels, and then we see another exponential growth period where it's eating the lactose. Usually, they're not the same growth rate. Usually one is a little bit slower than the other. Usually the lactose, it doesn't grow quite as fast on as the glucose. So just know that in real life, these curves would not be identical to each other. One would be a little slower than the other. I saw this a lot in my experiments. We deleted all the pumps and then put them in a starvation stress, and my graphs kept doing this over and over and over, because what they do is, yes, we want to eat the food that's available first, but bacteria can do something really cool where if they exhaust their food, they will start eating their waste products, like bunnies do. They'll eat their poop. So they're like, well, I have no more food, but this lactic acid that I made, I can break that down into glucose, I guess. And so they'll break it down and start eating their waste products, and that'll change their expression. Then they'll eat a different waste product, then a different one. And then eventually they're like, okay, now I ate everything. I can't eat any more of my waste, so now I'm gonna die. But we see a bunch of really interesting dioxic growth patterns because of that. Okay, now how do they know which source of carbon is present and which to use first? Because the glucose stuff is always expressed and it's the preferred method. So we have to do all of this work to turn on the lac operon, and the cell's lazy. It's not going to do that unless it has to. So if glucose is available and it has signals that it's getting enough food and it's happy, it won't turn all this on unless it needs to. All right, so let's talk about this catabolite repression for a moment here. The idea is that glucose is going to be brought in by that phosphotransferase system, the PTF system. And when that happens, it actually inhibits LAC-Y. So it prevents the cell from bringing any lactose in. It says, let's not waste energy eating this when we can eat the good stuff. So this is called inducer exclusion. So if glucose is coming in, this is not phosphorylated because all of the phosphate groups are being passed off to glucose. So it can actually repress LAC-Y and say, LAC-Y, don't bring any lactose in. Don't work right now. But if there's no glucose, then it's phosphorylated and it can't function to block LAC-Y. So LAC-Y gets turned on and lactose can come in. Does that make sense? Kind of cool. So the preferred carbon source also lowers our camp levels. If our camp levels are low, we're not going to turn on lots of expression. So we can block the entrance of the least desired food product and also we're not starving, so we don't send out starvation signals. Make sense? I hope like hearing it a second or third time, it's making a little bit more sense to you guys. I'm trying to take it from a big picture point of view about like what's happening, why are we doing this, rather than memorize this. All right. So CAMP, as I said, it will bind to CRP, then it will bind to our cell, cause the promoter to be bound more tightly by RNA polymerase, and we will create a lot of that's the idea there. So that's just another picture of it all for you, because like I said, more pictures, the better for bio. So this is just another kind of review of everything that I basically wrote up here. If lactose is absent, what happens? If lactose is present, what happens? And so just use that as kind of a review for yourself and kind of quiz yourself. Well, what happens if it's low lactose, high glucose, low glucose, high lactose, and try to figure out what happens. Any questions? Not too bad. Trust me, I've been where you're seated. It's not fun learning this stuff the first time. It's very stressful. But try to have a fun approach to it and it gets better, I promise. All right. So that is a catabolic system, and we'll talk more about this when we get into metabolism tomorrow, but catabolism is when we break molecules down, usually for food, like digestion. That's a catabolic process. An anabolic process is building up. So think about like anabolic steroids. They're used to put on more muscle. We're building more muscle. So an anabolic pathway is a biosynthetic pathway. We're trying to make things, like make DNA, make proteins now. So they have a different mechanism of control. They repress and activate in slightly different ways. So really, they only want to turn these on if the thing that they're trying to make is gone. If they have a lot of it, why would they want to keep making it? So we want to turn these types of genes on when we notice that the levels of this compound are too low. And so here we're going to talk about the tryptophan operon. It's like this, but I feel like it's actually a lot easier to understand. Normally I like to teach this one first because it makes a little bit more sense and it's fewer moving parts. But the book did it in the opposite way, so we're gonna just go with the book. All right. Usually, these genes are regulated by inactive repressors called aporepressors. And what they do, I'm sure you've heard this before, especially if you've taken like physiology or any course like that. This is very common in a lot of our digestive system, or not digestive, metabolism processes, and a lot of parts of our body where we have negative feedback loops. So what happens is we have a whole process of reactions that occur, and we'll have stopping points where they produce intermediate chemicals, and then we'll then do another reaction on that intermediate chemical to keep breaking it down or keep building it up. At some point, one of these intermediates or the end product often will come back and associate with the enzymes at the very beginning to either turn them on or turn them off. In this case, we're usually looking at turning it off. My end product, tryptophan in the case that we're going to talk about, when we have a lot of it, we don't need to be wasting energy to make more of it. So if I have a lot of it, some of it's going to come back and block the enzymes at the beginning from doing this whole process. That's the idea here. So the end of the line product is going to bind as a co-repressor. It is the ligand that binds to the repressor and causes it to bind the DNA and stop the whole pathway. That's the idea behind that. Make sense? Trying to say this as like simply as possible today because I know we're all a little brain dead, especially on a Monday. Okay, so let's get into the trip operon now. Maybe it'll make a little bit more sense. So the trip operon encodes 5 genes, all involved in making tryptophan, which is an amino acid. I know essential amino acid means something. It's an amino acid required to make our proteins. Now, tryptophan, just fun little fact, it's the least commonly used amino acid. So if you look at proteins, there's amino acids that constantly get used in high amounts. Tryptophan is like one of the less used ones. All right, so when our tryptophan levels are low, we're gonna start having a problem making proteins with tryptophan in them. So we're gonna need to turn on the whole process to make more tryptophan. And that's the idea here. The tryptophan itself is gonna act as a co-repressor. It will remove the repressor from the operon and we'll start making more tryptophan. Now, it's going to, sorry, wow, my brain today.
Okay, we're all there today.
Yeah. And not like the room messed up, it's like all crazy today. Yeah, so here we have our aporepressor, the TRP repressor. It's going to bind to tryptophan, and when it's bound, it blocks the whole process here. When there's low tryptophan, it's not going to bind, and the repressor can't bind and can't block this process, so we make our tryptophan. That's the main idea here. Do I have that over here? Yeah. Now, as I said, remember this is not just an on or off switch. Sometimes the repressor will fall off or fall on, and we'll have a little bit of it being made at all times.
The aporepressor was the TRP.
Yes, the aporepressor is the TRP repressor. The co-repressor is the tryptophan, and when they're together, we call it the holo enzyme or the holo repressor. Yeah. Holo just means it's fully bonded together with all its parts. So aporepressor, think A, once again in bio means not or against. So this is the repressor that's not working completely yet. It needs another part. It needs its co-repressor, its co-worker to come help it out, and that's the tryptophan. Good question. Thank you for having me clarify that for you. All right. Now, that is all of the tryptophan operon. So it's co-repressor, the aporepressor, is it on or off, basically, with repression. We're gonna add a little bit more to it now, but this is a different idea that we're getting into now. So we learned the lac operon and how it works. We learned the trip operon and how it works. Now we're gonna talk about, okay, but what if we have a little bit of tryptophan, but we want a little bit more? What if we have a little bit more and we wanna keep it there? What if we need to ramp it up a lot? How do we turn that rheostat? That's attenuation. Attenuation is the regulatory mechanism where translation is going to affect how we make our mRNA. So the path later on is going to kind of control how we make the mRNA, which is really weird to think about, but it will make sense when you see the pictures. Any questions before we dive into attenuation? This is pretty cool, actually. So, the leader sequence of the tryptophan operon is called the attenuator. So we have right here, before we get into the first gene of the operon, we have the leader TRPL sequence, which we call the attenuator. It does not encode a protein, it's a regulatory sequence, an untranslated region, basically. So as the mRNA is made, it's going to create 4 particular sequences that we're looking at. And we're just calling them sequence 1, 2, 3, and 4, just to make it easy. So let's dive into how this starts to work. Sequence 1 and 2 can bind to each other because they have complementary sequences to form what we call an attenuator stem loop. So 1 and 2 can bind together, and they like to bind together, and 3 and 4 then can bind together as well. But 2 and 3 can also bind together. So 1 and 2, and 3 and 4, but also sometimes 2 and 3 like to come together as well. 1 and 2 have very, very high affinity. So if 1, 2, and 3 are all available, 1 and 2 will come together first. They really want to be together. 2 and 3 will only come together if 1 is out of the picture. It's kind of like saying, I'm only going to be with you if you leave your wife, kind of thing. This is going to create different structures in the mRNA, which is going to affect how the ribosome binds to it and makes the protein. One thing I do want to say here, I think, no, I don't have it here yet. Right here at the beginning, we have codons for tryptophan. So we're going to try to start making the protein. If we have lots of tryptophan, we can make the protein here. If we don't have a lot of tryptophan, the ribosome is going to try to make this part of the protein and say, well, I don't have any amino acids to put here. What do I do now? So that's the whole idea of how we regulate this. So let's jump into that a little bit here. The ribosome binds, starts to make this sequence, and if it can bind, because our tryptophan levels are high, it will bind and just move right through sequence 1, because it has the tryptophan it needs to make it past that first hurdle. If that happens, 3 and 4 bind together, because 1 and 2 are occupied. So 3 and 4 bind together. When they do, they make this really awkward structure that kind of falls over onto the Shine-Dalgarno site for the rest of the operon. So this is where, or I'm not the Shine-Dalgarno site, I'm sorry, the promoter for the rest of the operon. So the RNA polymerase is trying to make more mRNA here, but it gets smacked on the head by this 3-4 loop structure. And as a result, it causes the polymerase to fall off, and we don't keep making the protein. Because it's saying, I have a lot of tryptophan. You don't need to keep making this, so stop. So the ribosome can move straight through. It causes 3 and 4 to bind. They smack off the polymerase, so we don't keep making more and more mRNA. So we don't make more protein, we don't make more tryptophan, because we have enough of it. Does that make sense? I think it'll make more sense when we talk about the next thing too. So let's go on to part 2 here. This is just the words that explain all that. Okay, let's talk about low tryptophan levels now. When it's low, it's going to try to make the protein here. It's going to bind, it's going to start the process of translation, but it's going to get to the 2 codons of tryptophan, and it's going to say, I don't have this amino acid, and the whole ribosome is going to stall or pause because it doesn't have anything to add there. When that happens, it hasn't moved forward enough yet. Now the wife is out of the picture and 2 and 3 can get together. So they love to get together. As soon as they're available, 3 says, screw you, 4, I'm going to be with 2. And they immediately bind. And now this doesn't cause that weird 3-4 loop structure to interfere with the RNA polymerase. So the polymerase can just keep going along its job and make more mRNA, which will eventually lead to more proteins, which will then lead to more tryptophan. So it's saying when we have low tryptophan levels, we don't want to interfere with the polymerase. We want it to keep going so we can make more tryptophan. That's the idea there. When we have a lot, we want to stop it from being copied. And this works as a rheostat where we can say, okay, I don't want to go straight from 1 to 10, I want to go to 2 or 3, because depending on how much tryptophan is already available, we might be able to make some of it, but if it gets lower and lower and lower, we'll make more and more and more. So it has its own control system built in because it's all based on whether tryptophan is available to block or allow this whole process to occur. Does that make sense? It's kind of cool, right? It's like it's happening in the bacteria in your gut right now. That's weird. I don't know. Okay, we're gonna keep going then. And this is just the words that break that all down. So you can use that to study from. Because I know sometimes I need the words written down too. Now, moving away from our operons and repressors and activators and all that, we're going to talk about other methods that we control expression. And one of them is with these alternative sigma factors that we've already kind of mentioned. We have, in E. coli at least, sigma 70. That's the housekeeping one. It's the one that's most often turned on. It's controlling all the genes that we need for a normal happy cell. But then there's other ones like sigma 32 that turn on when we've got heat shock. So now all of a sudden the bacteria gets put into boiling water and they're like, we're gonna die. Sigma 70, we don't care about you because these are not normal conditions. Turn on the heat shock response. And these alternative sigma factors will turn on a new set of genes that help the cell survive this difficult situation they've been put in. So, for example, we have RPOS. This particular sigma factor controls what we call a different regulon, a different set of genes. That includes things like acid resistance, antioxidant properties, base resistance, more antioxidant properties, and we turn this one on when we're starving. So usually when the cell is starving, it's eaten all of its food and it's made a lot of waste, and bacterial waste is very, very acidic. So we're starving, that means we're probably sitting in a lot of our disgusting acidic waste products. Let's protect ourselves from that. We're gonna start experiencing a lot of oxidative stress because we're trying to do a lot of metabolism and we're making a lot of ROS as a result. So let's protect ourselves from that. We're starving, so we aren't taking good care of ourselves anymore. So maybe we're gonna have more base mutations. Let's protect ourselves from that. So the housekeeping sigma factor gets turned off, and this starvation sigma factor gets turned on in these cases. And it just controls all these different genes all together are called the regulon. The set of genes that are regulated by that one sigma factor. Does that make sense? We're not gonna get too deep into this stuff, but I don't wanna talk about this. We're gonna cut it out of the exam. This is one thing we don't need to talk about. I want you to understand the idea of a sigma factor right now. Don't worry about this. We're good. All right. But we do need to understand there are also anti-sigma factors, because it's never just, oh, well, we made this one thing and we're done there. We want control over every little aspect of everything. This bacteria has been hurt before. It needs to control every aspect of its life now. We'll get hurt again. So, these anti-sigma factors are proteins, and they basically interact with all of the different sigma factors to inactivate them. Instead of binding directly to the sigma factor, they're going to block their access to the RNA polymerase. So they're going to say, sigma factor S, we're not starving. I'm going to block you from being able to turn on your starvation genes. But I'm not going to block sigma 70. It can do its job because we're happy right now. So just these anti-sigma factors are proteins that inhibit sigma factors. There's also anti-sigma factors. Just know that. You don't have to know more than that. And I'm sure there's probably anti-sigma factors we just haven't discovered. There's a lot of overlap in the cells. They make a lot of redundant things because in the end, they don't want to die. Same with our cells. It's much better to have 3 backup systems than only one and have that backup system fail and then you die. Especially if you're a single cell and this is your whole life. You don't have other cells in your body that can pick up the slack. Alrighty. We can also regulate our RNA directly. And this stuff is really cool. I'm not going to give it the time that the book did. I'm not going to give it the detail that we probably should in this class. But if you like this stuff, definitely take BACcheck. You'll learn some really cool stuff in there. That's where a lot of chemical engineering is happening, and we're creating RNAs that can act as like circuits to turn things on and off in the cell when we want them to. So we can give the cell a certain food product, and it'll turn on this circuit that turns on all these other genes that we want it to turn on. So it's like us controlling the cell. There's a lot of really cool stuff out there. But we're going to talk about a few of them that naturally evolved. These circuits are things that we make. An example of one. You guys have all seen Jurassic Park, right? The idea behind Jurassic Park is they thought they were very smart. And they're like, we're going to make all these dinosaurs and set them out on this island. Well, what if they get off the island? Oh, well, that's okay. We put in the lysine contingency. We have to feed them lysine or they die because we've blocked their ability to make lysine. That's an RNA switch. That's an RNA circuit that we created in real life now, basically. So it's really cool stuff. If you like that stuff and you're enjoying this genetic stuff, take BackGen. It's really cool.
You're saying that's in Jurassic.
The original Jurassic Park.
Don't they just like mutate or like adapt?
Yeah, that's the idea with Ian Malcolm being like, life finds a way. It's gonna mutate.
I'm uncultured. It's a slow morning.
It is a slow morning. That's why I'm trying to let's just talk about Jurassic Park for a minute. Let's breathe for a minute. Okay. So let's jump into this. We can have RNA sequences that don't code for a protein. They actually exist to regulate other mRNAs. Remember, it's all based on base pairing. So if we make a code that base pairs to an mRNA and they bond together, that mRNA can't be accessed by a ribosome now, so it can't be turned into a protein. So it's one way to regulate what mRNAs get turned on into proteins by the ribosomes and which ones don't. Some of them will actually be able to break down other RNAs and cause them to degrade because RNA has that enzyme aspect to it as well. So let's get into some of these examples here. We talked about one with attenuation already, but we're going to get into riboswitches here. And these are just sequences that they will fold into three-dimensional shapes, and that's going to affect their ability to bind to different metabolites and kind of sense what's going on in the cell. So they will create these really cool structures, and sometimes these structures can bind a metabolite or a ligand, and that's going to affect how they bind to other RNAs to turn them on or off, basically. I don't need you to know more than that for the exam. If this is on the exam at all, this stuff, it's going to be multiple choice questions. I'm not going to ask you a short answer about this stuff. All right. The only thing I want to say about this is that they exist, so let me just get into them a little bit. We have these siRNAs, they're small RNA molecules, and they go around and just inactivate other RNAs. Cis antisense RNAs are kind of the same idea, except they come from the non-template code rather than the template code. So they're just another way of inactivating other mRNAs. That's really all I need you to know from those for now. And if you want to know more, the book tells you there's more information here about the siRNAs. I was going to get into all this with you. We don't exactly have time, and like I said, I'd rather you focus on learning the operons than us getting into that. So, second messengers we are going to talk about here. We talked about CAMP as an important second messenger, but these are just small molecules that they're going to help regulate all the functions of the cell. They tell the cell what's going on and will bind to different proteins to regulate the DNA. We talked about one of these a while ago, guanosine tetraphosphate, PPGPP. In the past, it was also called Magic Spot, which is a really weird name. Why, I don't know. That's way before my time. But this guy allows the cell to sense changes in nutrient availability. And it's going to help the cell respond to starvation versus lots of food stressors. This is called the stringent response when we have nutrient scarcity. So many ribosomes are going to be idle. When that happens, it triggers the synthesis of PPGPP. So idling, like ribosomes that have nothing to do causes more PPGPP to be made, saying, hey, we're not making stuff here, we're starving here. And the PPGPP causes the cell to start turning on a different set of genes. And those will help it respond to the stress of starvation or not having enough building materials, basically. Any questions about that? Okay. There's also cyclic di-GMP or C-di-GMP. This one's kind of cool. It controls the turning from a planktonic bacteria to a biofilm bacteria. So this is the second messenger that tells the cell, hey, we want to transition here, And basically, if we want to swim, we have low CDGMP, diGMP, sorry. If we want to swim, we have a low amount of this. So that favors the formation of flagella. When we have a high amount of it, it's going to block our flagella genes. It's going to block all of our motility genes, saying stop swimming. Instead, I want you to start making genes to make biofilms. So that just helps the cell regulate when it needs to switch between those two states. That's what I want you to get from that one. And these are all just secondary messengers that accumulate based on the environmental conditions of the cell. All right, that is it for today. Let me stop the recording here, and then I want to share one thing with you guys.